Thank you, everyone. Oh, I'm not on my right side. Oh, there we go. That's better. So I wanted to start, as Mark mentioned earlier, I didn't feel like we'd had quite enough weather charts up yet, so let's have a few more. And I wanted to sort of pick out the spring and winter average temperatures over the last few years. And really, this is just to point out that we don't tend to have an average year anymore. I think we all knew that. But in particular, springs are perhaps getting a little warmer and a little wetter, and that is changing our disease pressures as well. So we've had a few examples of this that we could perhaps pick out on. Perhaps beet moth. That was something that I don't think we'd ever really seen. It came in quite suddenly. I particularly noticed it in 2022. That was the first year when I was doing my PhD. I ventured out into the field for the first time, left the safety of the lab. Field trial was completely decimated by those particular moths, much to my dismay. Another example, Cercospora. When I first started at BBRO, if we had an example of Cercospora into the plant clinic, it was really quite exciting. It was quite a novelty, whereas now we've got whole BBRO projects dedicated and Spornado profile right across the country trying to monitor its arrival. But it's not just new diseases that are changing with climate change. We've also got some things that perhaps we are familiar with but are being more favoured by these conditions. And for that, I'd like to propose to you Rhizomania, favoured by warm, wet springs and perhaps bubbling its head about. And it's for that reason I want to talk about revisiting Rhizomania over the next few moments. And to do that, I'm going to use a real plant clinic case. We're just going to go through and hopefully show you the importance of detailed diagnostics in these times of sort of changing disease pressures. Okay, so I know we all know where this is going. I know we know where the end is, but bear with me. Plant clinic case came into the lab. We'd got yellow leaves, really fluorescent in colour. I'm not talking the orangey colour that you might see with virus yellows. These were really fluorescent yellow, quite different. Lots of lateral roots, but no cysts. This wasn't BCN, but lots of lateral roots. And the roots had a quite unique shape, almost wine glass shape, constricted. Shameless plug at this point for the plant clinic. That QR code will take you to the new JOT form system. I appreciate the form can be a little bit of a pain to fill out, but the more detail you give us, the more detail we have to work with and the better answer we can ultimately give you. This case, we did have some really good pictures, some really good information, and that enabled us to start thinking Rhizomania. What is Rhizomania? It's a virus. So we're beet necrotic yellow vein virus. It's an RNA virus. We'll come back to that in a moment. And it's transmitted by a parasitic protist, something that we might once have called a fungi. That's not quite true anymore, called Polymyxobetae. And it's that organism that's favoured by the warm, wet conditions. Those warm, wet soils will enable it to infect your roots. And if it's carrying the virus, you then get your Rhizomania infection. The Polymyxobetae lives in the soil, like I say, and can survive for very long periods of time as resting spores, decades. So once a field is infected, you have Rhizomania in that field virtually for perpetuity. I am very conscious in the audience that those of you who were farming in the 80s are all too aware of Rhizomania. But, and apologies to Mark, who I know worked on this in his early career, that was a good 30 years ago. So for the benefit of the youngsters in the audience, let's have a little recap of our timeline. So Rhizomania was first identified in Italy back in 1952. And it was really starting to cause some major problems. It spread consistently across the continent and further afield. And we were watching it as an industry, growing increasingly concerned, and tried our best to stop it reaching our shores. So we had legislation in place, things like limiting imports and exports of sugar beet roots and soil. But ultimately, it did make it to Suffolk in 1987. But thankfully, breeding efforts and like Mark was saying earlier with SBR, knowledge of the continent meant that we had our first Rhizomania variety introduced in 1997. As an aside, that was also the first year it was reported in the Newark factory area. Variety adoption was really quite successful, and by 2013, the entire UK crop was sown with partially resistant Rhizomania varieties, and the disease had been declared endemic at that point, and your legislation controls dropped. I say that the entire crop was sown with partially resistant varieties, so much so that now it's almost hiding in plain sight up there in the top left of your recommended list. It almost goes unnoticed that all your varieties are RZ1. But like I said earlier, we're dealing here with an RNA virus, and viruses do like to mutate. They like to change and evade us, and that is exactly what happened. The story is not quite so simple, because in 2007, we had some examples of disease symptoms showing in crops that were RZ1 genotypes. A series of genetic testing led to the identification of the AYPR strain of Rhizomania. If we're really drilling down into the detail, AYPR, that comes about due to the really tiny changes in the RNA virus's genetic code. RNA, third fragment of the virus, changes to an alanine, tyrosine, proline, and arginine amino acid protein building block, and it is that tiny change which allows it to evade the RZ1 resistance gene. I admit that's possibly a detail too far, but that change has a big impact. Fortunately, the additional resistance gene, RZ2, had been identified, and combining those two resistance genes in the sugar beet variety meant that we could keep crops protected even from an AYPR strain of the virus. So back to our plant clinic case. From the information we were given, we knew it was an RZ1 variety, but we're still seeing symptoms. That really gave us in the lab sort of two options. Option one, this is a case of AYPR, but we didn't know was in the field. Or option two, this is something new and a bit scary. Another option, another strain has developed that we don't know about. And that's where I come into the story. So I finished my PhD back in February last year. And one of my first tasks was to develop the Rhizomania diagnostics in-house at BBRO, make sure that we are able to identify these strains, and in particular, identify any novel diseases coming in. Traditionally, we've used an antibody-based method. That's a really good technique. ELISA is superb for getting through a lot of samples. It's very high throughput and can tell us whether the virus BMYVV is present or absent. But it can't do strain identification. For that, I had to bring in a slightly different approach. This isn't new equipment, it's not new chemistry, but PCR and gel electrophoresis is really specific. We can really drill down into the exact strains of the virus present. Not high throughput. I can't do a whole field scale analysis, but that's not really what this is about. This is getting absolute detailed diagnostics to identify if we have a serious problem or something that we can manage with varieties. And that's exactly what we did in this case. So by testing, using that new technology, we were able to identify that in this particular plant clinic case, we had the AYPR strain of Rhizomania present in the field that we previously didn't know about. And in a way, this is a good news story because we could suggest that in the future, we use an RZ1 and 2 variety in this field and hopefully limit the problem. Wasn't anything new or scary. So for the next few moments, I'd like to just think about what other diseases might be bubbling along in the background that might benefit from more detailed diagnostics. And I'd like to pitch to you blackleg. So seedling diseases, as some of you may know, infect normally shortly after emergence and are generally controlled by our tachygyrin seed treatments that we have available. But plant clinic cases have been creeping up over the last few years, particularly in 2023, where the entirety of the year's plant clinic cases, it was about 23% of them were seedling disease related. like I say, those warm, wet springs favouring the fungal development in the soil. Here you can see on the picture there, oh, have I got a pointer? Perfect. So if I make this a little bit bigger, we've got a classic example here of Aphanomyces infection. So you've got the leaves aren't wilted, but the root is blackened, giving it that classic blackleg name. And generally our diagnostics are limited to symptoms. But the problem is, although Aphanomyces has some really classic and easily identifiable symptoms, the other causes of blackleg don't. So this is a complex disease caused by multiple organisms. You've got Pythium, Rhizoctonia, and Phoma, all potential causal agents. And at the moment, we don't have diagnostic tests to identify them. But perhaps in the future, that might be something we need to think about with changing chemistries, changing disease pressures, and think about that a little bit more. But I'd like to finish with a final plug for the BBRO Plant Clinic. And in the short term, hopefully it gives you the answer to the specific question you're asking and your specific problem. But it does have a longer term effect. It's through the plant clinic that we see what's going on and can potentially identify where we need to be focusing our efforts and bring you those, focus our R&D efforts on what will give you the biggest impact. And with that, I think we're continuing the disease theme with Alistair.